I'll do that.
Oops.
Mike,
Yes,
can you hear me?
yes, I
Yeah.
can.
Okay. Hi, it's Elliot. Just wanted to say hello.
Hey, congratulations, Elliot, on PNAS.
Thank you.
Or in the National Academy, I should say. Yeah.
Yes, and Elliot, I should both say to you, Mike, welcome back to WashU. We missed you here for a while. So glad you're back.
For some reason, it's not taking my picture, but I think you're all the benefiters from that.
So, Mike, tell as the gang gathers around here, tell us about life back in the United States. How is it being a Texan and an American?
Well, I must admit, Texas is certainly different than WashU and certainly different than New York. The interesting thing here is that this is basically a vacation town, a resort town, and so the medical school is sort of although it predated all of that, is sort of isolated out here, but that also affords us the possibility of concentrating on things, and that's what we're doing right now, focusing on trying to get this cancer treatment into the clinics. So, yeah, it's been very good for that. So,
yeah. So is your lab all set up now, Mike?
Lab's pretty much set up, and we're proceeding now with a number of new areas, primarily focusing on mitochondria and their role in both senescence and in cancer. This ER mitochondrial stress pathway, as I'll emphasize today, appears to be a weak point in the transformed cells. So,
so do you study in vivo, like inside the cell, the mitochondria, or do you purify it?
this is what we're starting on, using a variety of various calcium-sensitive dyes, potential dyes, and so forth, to look at the effects of the ultrasound as we are irradiating the cells.
Do you still have affiliation with Columbia? Do you still have a lab there or anything?
I'm emeritus there, but you know, I'm not active emeritus there. So I don't have a lab anymore and so forth.
Okay. Yeah. And how about NUS? Do you have any students there?
Yes, I have a couple students, joint students still there, and sort of am a co-PI on a number of grants and so forth there. So, yeah.
Mike, I actually got one of your former postdocs, Brian Doss. It's not in my,
Oh, he's working with you now.
Yes.
So that's good.
Very good.
Bryant is very, very hard worker and very solid engineering.
Yeah.
Small mechanobiology world. Where can he go?
That's right.
Mike, you have been in the
Illinois.
field for a long time. Do you know? Do you have a sense of how many active researchers in the field of mechanobiology now? Broadly defined.
Well, it depends on how you define it always. Yeah. And I see a lot of papers and abstracts emphasizing mechanobiology, and they're looking at various aspects of it. So I mean, I think that it certainly is a catchword, and there's a lot of interest in it. I think there's less understanding of what really is involved, but that will come.
So in your definition, how would you define the field of mechanobiology?
Well, I think it's getting to, we want to get to molecular mechanisms and the steps that are involved, rather than put in a stimulus and wait a day and then find out what is the response. Okay. There are steps, thousands of steps in between. And for us really to understand these processes, you need to understand how those steps actually work what are the feedback mechanisms, and it is not a trivial issue to make a deterministic process out of a stochastic system. And that's the real problem.
So the mechano part comes from somehow force being involved. Is that your thinking?
Well, you have to shape the organism. And the real issue in biology is the biodiversity that's out there. The various different shapes will define the function of those organisms. And that's what has happened throughout evolution to enable those organisms to survive and to carve out a niche.
Mike, on my mind, you introduced mechanobiology to me. And that happened when we organized a workshop together called Cell as a Machine.
Right.
This was back in 2009. Very interesting workshop. I learned a lot. And after that, we invited you to come to University of Illinois for GM4 Summer Institute. And all these interactions significantly enriched my own research interest. So for that, I'd like to thank you for.
Well, thank you.
Yeah, this is wonderful, wonderful.
Jimmy, do you remember which year was it?
It was NSF in
correct.
Yeah, when we were young.
Right.
2008 or 2009, one of those years.
Yeah.
Right.
Yeah.
So Mike, you had your master's degree, I believe, on mathematics, right?
Pardon?
Your master's degree was in mathematics, or?
No, I never had a master's. I mean, I was an undergrad and I had a dual degree in chemistry and chemistry and mathematics.
Yeah, that's what I was thinking. Yeah. Okay.
Hey, Taher.
Yeah.
We have quite a few panelists here with us, and
yes.
would you mind to help introduce our panelists to the
audience?
audience?
Absolutely. Let's start. I don't know whether my screen is the same as everybody else's screen. I know quite a few. Maybe what we could do is I could just ask you to say where you are. I just mentioned the name. So Jimmy, you start.
Oh, Jimmy Xia. I'm co-editor-in-chief of EML. It's very nice to meet all of you. I'm a professor at Nanyang Technological University in Singapore.
Guy?
I'm Guy. I'm from Washington University in St. Louis. I'm the Center for Engineering Mechanobiology, and I'm doing my best to follow in Mike's footsteps here as a WashU mechanobiologist. And I have a lot of traveling to do to follow those footsteps.
Andrew, if you all
hi everybody.
Unmute,
Excuse me?
If all of you can unmute, then there's a
I see, yeah. So I'm Andrew Hall. I've actually met some of you before, but some of you I haven't met, so it's very nice to meet you all. I actually just started at the Mechanobiology Institute in Singapore just in January. So of course I'm very indebted to what Mike actually built here. So it's good to see him today.
Joy.
Okay, Tang?
Hi, hello everyone. My name is Tang Li. I'm a professor in the Department of Mechanical Engineering in the University of Maryland. I'm also one of the editors of Extreme Mechanics Letters. Great to have you, everyone here.
Great. Daphne?
Yes, hi, I'm Daphne Weiss. I'm in biomedical engineering at the Technion in Israel. And as a chemical engineer, I come in from the engineering and mechanics side of mechanobiology.
Jennifer?
Hi, everybody. I'm Jennifer Young. I'm also a new PI at the MBI, so very thankful to Mike for setting that up. and also with the biomedical engineering department at NUS. So nice to meet you all.
Great.
Looking forward.
Zhigang.
Zhigang Suo, Mechanics and Materials at Harvard, also a co-editor-in-chief of Extreme Mechanics Letters. Mike, thank you for coming.
My pleasure.
Elliot.
Elliot Nelson. I'm at Washington University. I am really retired, but I tag along with Guy Genin's group.
Sulain.
And we used to collaborate a long time ago.
We did indeed.
Sulain Zhang from Penn State. I'm also one of the editor of Extreme Mechanics Letters. Very nice to meet all of you guys here, and looking forward to Micro's talk.
Brian.
Okay.
Brian Dawes. You are muted.
Brian is a former postdoc of Mike's. I don't know whether I promoted him because he's now a postdoc in my group, a research fellow in my group.
Okay. Okay.
Not sure whether he wants to say something.
Okay, that's all right. Okay. All right. So that's all I have seen. All I see in my screen thing.
Yeah, go ahead back to the interview, and I guess I will welcome all the panelists to ask questions.
Yeah, but also Taher and Guy, I think I will rely on two of you to promote others to the panel.
It's a lot of power.
When you see familiar names, to promote them. Okay.
So Mike, I'd like to ask you kind of a big question about the field of mechanobiology and where it's going. So, so really the field of biochemistry really started making, about the time the field of biochemistry really started making true, undeniable impact. We could tell when that happened, and not just from the increase in the number of real cures that went out there, but from the sort of proliferation of all of the kind of fake nutraceutical industry and such. And so the broad question I'd like to ask is, where does mechanobiology stand, sort of in terms of overall impact, and also in terms of overall pervasiveness in society? When are we going to start having mechanobiological recommendations for health coming out on CNN? Are we close to that yet? Or where does the field stand?
I mean, I can't obviously predict those sorts of things. They're very culturally related. But for a long time, it's evident that, the doctor can tell you, I can give you a drug for this, or you can do an exercise. Okay. And the mechanics are becoming increasingly, popular in terms of the aging population, the benefits of walking, exercise, activity in that aging population. And we don't understand why that is and what are the, what's the basis of that. And so I think that there will be an increasing interest in this if our ultrasound therapy for cancer works as well as I hope. That will have impact as well. And, a lot of things in the aging process, in many disease processes, are mechanically driven and affected very much by mechanical activity of the patient. So I think that's a large area, relatively low cost. I mean, you compare ultrasound treatment of cancer with drug treatment of cancer, and there's no comparison in terms of relative cost. So
I think that gets back to Guy's question. If you can't put it in a bottle and sell it, you're at a big disadvantage.
Well, that may be the case. I mean, we formed a company, Mechanobiologics, to test this out, and we'll see what we can do. But I think there is a great future And it's only through understanding this. And the other issue that I think is evident now is that nanotechnology has enabled us to look in the subcellular level at these mechanical processes. And that will help us understand at least what is happening. And from that, we can build perhaps a better way to approach these problems.
Mike, I have seen a paper not too long ago. I think it was in one of the Eric Sahai's paper in Nature. So he outlined, I think, about 10 or 12 drugs under trial now for cancer related to mechanobiology. So either taking out the force from the CAFs or one way or the other, they're using the concepts of mechanobiology. And some of them have gone to trial 3 now. So,
right.
Elliot, to go back to your question, things are coming up in the bottle. Probably
well,
not next
obviously,
year.
yeah. Obviously, when we say that there are molecular mechanisms behind the mechanics, those are potentially druggable. The issue, I guess, is that when you look at the complicated nature of many of these processes, as in a multi-step, normal engineering process, how to build an airplane, et cetera, you understand that having just a change of altering one part often doesn't do the job of fixing the problem, because it's used over and over again and in a variety of different ways. And so I have some skepticism about the ability of a simple drug to meaningfully affect a larger process, a mechanical process.
So Mike, you're going to show us how stress will kill cancer cells.
Right.
Do you see any potential impact of mechanobiology on coronavirus? It's a pity not to talk about the pandemic right now.
We tried some things with that in a preliminary way, and there wasn't much of an effect of either on infectivity or on other aspects. But what was evident was that there was a much greater effect of the virus on aged cells. So we had some senescent cells and we tried infecting those, and those were much more susceptible. We really didn't follow up with trying to reverse the senescence, which we can do mechanically to see what effect that might have.
Can I ask you a question on that area? So did you guys all see that paper from Uri Manur's lab in Circulation Research showing that the spike protein from SARS-CoV-2 binds to the ACE2 receptor with kinetics that somehow end up going through a long cascade that end up interfering with mitochondria, which then subsequently interfere with the vascular endothelial cells with the function of various clotting agents, and leading to perhaps leading to some of these pathologies. So the question is, it's kind of broadly getting back to where you're talking about a moment ago, Mike, what's the future looking like, if you could forecast for us? What's the state of the art in your mind of being able to come up with ways to interrupt mechanical pathways that intertwine so closly with chemical pathways? And so if there is going to be a mechanobiological role, this is one target, but it requires something that's kind of in between the chemistry and the mechanics.
Sure. Well, obviously, I don't presume to answer that question at this time. The issue is more of approach and to really see what mechanical manipulations will affect the process, the overall process, and then how those might be incorporated into a treatment, et cetera, down the road. But I think we have a lot to learn about particularly mitochondria. They seem to be this ER mitochondrial junction stress pathway is really at the heart of the tumor cell killing by mechanical manipulation, and that appears to be altered when cells are transformed. What that means, I can't say at this point, but certainly the internal workings of the cell are really quite different when you have a transformed cell versus a normal cell.
That raises another pathway to think about this, which is not only using mechanics therapeutically, but using mechanics as a way to screen for the effects of therapeutics. And I know a number of people are working on that. For example, here at Washington University, Mike Greenberg is using mechanical assays to look at mutational disorders of heart cells in tissue constructs, heart tissue constructs. And that kind of thing is, I think, becoming more and more widespread.
Well, it's an integrated whole. I mean, and in other words, that the mechanics is intimately involved in the biochemistry. I mean, clearly for myosin, that's everyone believes that, but for many other aspects as well. So.
There are a couple of new people just joined. Maybe.
Robin,
yeah.
Okay.
Hi, everyone.
Introduce yourself quickly.
So please introduce yourself. Let everyone know where you're sitting today. And
okay. I'm Hagai Wolfenson, ex postdoc of Mike at Columbia. Now I have my own lab at the Technion in Israel, at Haifa, at the Faculty of Medicine. So also following up on my postdoctoral work, but from different angles.
Thank you. Rubin?
This is Robin Bai. I was a PhD student at of Zhigang a few years ago. Now assistant professor at Northeastern University, Boston. It's a very exciting topic. Look forward to it.
Ning. Ning, you have to unmute.
Oh, yes. I'm on here. Hi, Mike. Hi, how are you? Hi, everybody. Hi, Jimmy. Wow, it's been a long time.
So I will introduce Ning then.
Hi, Guy. Wow, Guy always has a beautiful, you always have a beautiful smile.
So Ning is a professor in mechanical science and engineering, University of Illinois, and he's sitting in front of a nice spring day.
Yeah, it's our library. My background is our library.
Yeah.
I'm Rinder as well.
Hey, good morning, guys. Thank you for organizing this exciting talk. I'm Amrindranath in mechanical engineering at Virginia Tech. My research is mostly focused on looking at cell fiber interactions, and we try and mimic the ECM to a very high precision. I'm looking forward to the talk, Mike. Thank you.
Good to see you again.
Good to see you, Jimmy.
And then just joined Shirish Jain.
Hey, hi to everyone, and hello, Mike. It's so nice to see you back after long. So yeah, I did my PhD in Mechanobiology Institute, Singapore, and I graduated in 2019, very recently, and currently doing a postdoc in Institute Pasteur. I'm again following the recent work of Mike and trying to see in 3D spheroids the response of external mechanical stimulus, not going on the line of ultrasound, but really external mechanical stimulus. I'm looking forward to the exciting conversation by all the panelists.
Denise? has joined.
Yeah, who is this Dennis Disher guy? Dennis, can you say where you are?
Yes, we can see you now, Dennis.
Okay.
We're just going to the introductions, Dennis, just to say where you are now. Yeah.
Hi.
Dennis has a camera this time.
Yes, a camera. Yeah. I do have one sometimes. Yeah. Yeah. Thanks for including. Yeah. Sorry, I had to join late, but busy with things. So look forward to Mike's exciting talk, and good to see all of you. I guess we're starting still at 10 a.m. Yeah.
Yeah. Maybe one last.
Okay, we should probably. Sorry, Taher.
Yeah. Go ahead, Jimmy.
Maybe let me start by sharing some slides. Let's see. Do you see it now?
Yes.
Okay. Since this is the first or inaugural lecture for EML Webinar Season 2, we decided to take a few minutes, maybe a couple minutes, to make one announcement and plus a short report on Season 1 and so on. Why don't I? Maybe let me redo it.
While you're doing that,
to point out that papers go through EML even faster than slides do. It's amazing how fast they process.
Okay, so the announcement I want to make is the following. This is the organizational structure of EML, and on the management side, Rebecca Kaboon from Elsevier is our executive publisher. Zhigang Suo, John Rogers, and myself are editors or co-editors in chief. And the good news is the former associate editors, 5 of them, Katya Bataldi, Hanjin Jiang, Teng Li, Matty City, and Sulin Zhang, they have been promoted to editors. So please join me to congratulate them. And last year we had Season 1 of EML Webinar series. I'll ask Zhigang to give us a short report on that.
All right. So thank you, Jimmy. Last year we just because the pandemic was the first time in my lifetime anyway. So we just did something. It turned out to be extraordinarily well receptive. We had a total of 32 webinars, more than 100 hours recordings. They're all on YouTube, and this many views, just collectively. I don't recall any conference has achieved this kind of enormous numbers. Yeah, Jimmy, next. Yeah, also this is Season 1. Again, all these webinars, all 32 of them are posted online. Some of them probably will be watched after a decade or two. They're so basic, so fundamental.
Okay, thank you very much, Zhigang. And now we're starting Season 2. Zhigang actually wrote a little perspective article called "To Read is Human, to Watch Divine." That was published in Matter, Volume 3, back in July 1st, 2020. Certainly, I encourage all of you to look into Zhigang's souls to see what he was thinking about when he proposed to have the EML webinar series. Season 2 is very similar to Season 1. We have decided to have reduced frequency because every week is a little too much. But we emphasize on interdisciplinarity. And we have some new tools that we can work with, QR codes and so on. And we also have established an independent EML Webinar Zoom account. So we're all researchers. We want to do experiment. And we decided to do a social experiment. I'm going to let Tom to explain the social experiment.
Okay, great. Thank you, Jimmy and the Jigang, for the review of season 1. And as you can see from the previous slide, and the season 1 of EML Webinar has been a great success. And we would like to thank all our speakers, discussion leaders, and of course, every one of you, the friends of EML Webinar, for your steady support and help, without which this success would not be possible. And in particular, we would like to thank the Harvard IT for their technical support for the EML Webinar Season 1. I remember one of the EML webinar friend shared during one of the live session that EML webinar has become my weekly ritual in this special year of 2020. I think he was not alone. So today we announced this launch of the EML Webinar Season 2. And to keep the EML Webinar Season 2 going, we actually, in this season, we will need some baseline funding to cover the basic operational expenses. As Jimmy mentioned, these basic operational expenses include, but not limited to, for example, right now, what we are using now is an independent EML Webinar Zoom account for the webinar service. And then you probably notice that as every individual EML webinar, this is recorded and stored in the cloud. And there is no free lunch. We need to pay for that. And there are also some related expenses to provide access to the existing EML webinar recordings in the years to come. So I have this question for every one of you right now. Either you are in the live Zoom meeting or watching the YouTube live, and also, and this season we start to stream lively in WeChat as well. And so far, you know what, we have more than 3,000 people watch this live in WeChat live stream. So I have a question for every one of you. So to keep EML Webinar Season 2 a fun place, just like now, for us to meet and chat with and learn from each other, would you be able to contribute to this incredible cause? So, so to make this simple, so we have this QR code on the screen. We started this GoFundMe initiative, and we set the goal of $3,000 US. We already have $100 US donated. So if you have a yes to my question, so very simple, take out your smartphone, scan the QR code right now, or you take a screencast and you can scan it later on. So the best time to plant the trees 10 years ago, and the best time to donate for EML webinar is right now.
Okay, thank you very much, Tom. Let me stop sharing over here. Mike, can I ask you to load your slides?
Sure.
Meanwhile, Taher, it's all yours.
Okay, very good. It's an absolute honor and, of course, the greatest pleasure to have Mike with us. So Mike does not need any introduction. He almost nurtured all of us in mechanobiology. But for the sake of introduction, I have to say that he is now the Robert Welch Distinguished University Chair in Chemistry at the University of Texas Medical Branch at Galveston. Before that, he was the Director of Mechanobiology Institute at the National University of Singapore, I believe for more than 10 years. And then before that, he was the Department Chair in Biological Sciences at Columbia University. He got his bachelor's in 1968, PhD in 1972 from Caltech. He got so many awards. I was looking at many of them, and then I realized that maybe I would take all his time. The most ones that I felt, he is listed as one of the top 20 most influential medical researchers alive today. And he got the Mesri Prize in Biomedical Science and Lasker Award in Basic Biomedical Sciences 2012 for his discoveries concerning cytoskeletal motor proteins, machines that move cargos within cells, contract muscles, and enable cell movements. He also received the Humble Senior Fellowship. And his recent studies defined the major contributions or components of matrix rigidity sensor, which is missing in the tumor cells that makes them transformed. So hopefully one day we will have a cancer solution through mechanobiology. These are all things that he has published over many papers. But the most important thing is every time we have come across with Mike, at least myself, learned something new. And his papers are the ones from which we have all benefited and nourished ourselves. So with that, I will let the floor to Mike. My request is if you can turn off your, mute yourself, just in case to avoid any background noise, and also the videos. Once Mike's talk is finished, then please come back. And from this point on, Guy will moderate the session. So Mike, the floor is yours.
Okay, thank you. You can hear me fine?
Yes, Mike.
Good. So today I was going to talk about our recent results on mechanical stresses as a way of killing tumor cells. We think it's fundamentally due to the difference in the transformed state versus the normal state of cells, and I'll explain a lot of what I mean by that. By way of background, in terms of mechanobiology, the real major question here is how cells create the form of the organism. And this picture of a bird of paradise that I was fortunate enough to be able to take in New Guinea, Papua New Guinea, illustrates the complexity of the problem. And the issue fundamentally is that within cells, things are driven by diffusion. All processes are diffusion driven. And yet, even though diffusion is a stochastic process, you have a deterministic outcome in biology. Organisms with the same genome often look very similar, or if not alike, and twins, identical twins, appear remarkably alike, even though all those decisions that were made in terms of the mechanical forming of those organisms were done separately over time. And I don't feel we have a good way yet of understanding how this diffusion-driven system can make those deterministic decisions. So that's a major challenge. Certainly when we look in detail at mechanical processes, they seem to go through a cycle of moving, testing, whether that's sufficient, and then reactivation movement. So it's a reiterative process and not like you or me going from here to there where we get in the automobile drive and we stop. It's not a continuous process, but in fact, a cycle of various testing steps intertwined with the actual process of movement and creation of forces in there. The other thing that we should keep in mind is that evolution has selected robust life forms. The classic example of this, which has come from the New York Times article, is that tardigrades have survived 5 mass extinctions. They can survive boiling, freezing, desiccation in outer space. So they are robust. And when you look at the issue of robust functions, and engineering robust functions is a very important aspect of engineering now. This is from a review by Nam Soo, who was at MIT, then moved to KAIST in Korea. compartmentalization of functions is one of the important issues. Modular components, functions are largely digital, not analog. They're on or off. Term limits, you don't do something very long before you turn it off. Cyclic in nature and variations on important functions. The one that I'm going to emphasize a lot is phases or linked functions with sharp transitions. And this is talking about the normal versus the transformed state today, and I'll emphasize that a lot. The other aspect of biology, which is remarkable, is that not only is it programmed into the organism to form the proper shape of that organism, and there's obviously biodiversity emphasizes how many variations on that theme there are, but you can have regeneration. The champions of regeneration are the salamanders and axolotls who can regenerate a total limb. And I think one of the features of that is that common molecular mechanisms exist for both regeneration and for healing. And microRNA 21, which I'll come back to again, seems to be increased not only in axolotl limb regeneration, but in wound healing, in brain, skin, liver, etc., etc. And it knocks down various components that block growth, and so enables growth and consequently regeneration. And it is indeed that issue which is important in cancer, because cancer is basically regeneration gone wrong. So when you have an injury, the cell wants to remove the block to growth, which involves interleukins, microRNAs, and a number of other factors, local tissue growth signals. You get tissue growth, and then you repair and consolidate and go back to a normal behavior most of the time. However, in certain instances, you don't restore the system properly, and consequently you have continued growth, which becomes a tumor. And most tumors are benign, we all have them, and it's only when they have these enabling adaptations do you get major tumor growth and metastasis and cancer. But common in all of those cancers is this removal of the block to growth. And indeed, if we can find something that will target that aspect of the system, we might be able to treat many more cancers with that treatment than focusing on those details of the enabling adaptations. Now, it seems that from the earliest days of cancer biology, there was something altered about rigidity sensing. And the cancer cells that they tried to culture in vitro would grow on soft agar, whereas normal cells from the same tissue would not. And hence the idea of transformation and transformed growth implied that the cells ignored aspects of that microenvironment that enabled them to grow, as you might expect for a repair and injury type of situation. Now, from the work of Dennis, it's clear that not only is growth on soft surfaces important, or ignoring or rigidity sensing as important in cancer, but also in differentiation. So when you put mesenchymal stem cells on soft versus rigid surfaces, they went down different differentiation pathways, soft going to brain, and rigid going to bone, and intermediate going to muscle, implying that that was an important decision-making step in the overall process of differentiation. When we've looked at this in detail, I think I made the wrong order here. We saw that the cells sense rigidity at a very early time. So these are mesenchymal stem cells put on tall, i.e. soft pillars versus short or rigid pillars. And we see the morphology of those cells as they spread indicates that they are sensing at early times the rigidity of that substrate. If we think about this and back this up here, and in terms of what is going on in those cells, we find that there are really multiple levels of organization in cells. At the really atomic level, we are understanding the structure of many of the proteins and how those proteins interact intimately to do their localized functions. At the other extreme, we see how the overall cells behave, but we know relatively little about the mesoscale, i.e. from 20 to 2000 nanometers, where you have various protein complexes. And what I'll be talking about today is the rigidity sensor, which is a 2-micron machine, which is a module that assembles and disassembles in about a minute. We see here this cell spreading on the surface, testing the rigidity as it spreads every 24 seconds. And like this automobile engine, thousands of components are involved. Force, position, and timing are critically involved in both of those processes. And indeed, we find rigidity sensors present in a variety of different backgrounds, just as you can take that automobile engine and put it in a number of different car bodies. It will work the same for the rigidity sensor. And further, as we'll emphasize in cancer, removal of a single part is often sufficient to block function of a modular machine like that. Okay. So we've looked at this in the process of cell spreading, and here I want to emphasize that I think it's time that the engineering community get more involved in the molecular processes rather than adding agent X or treatment Y and then seeing what the outcome is days or hours later. I think it's important to start now breaking these apart in steps as you would the process of forming a plane or creating any complex device. And then you can understand indeed how that process works. Here we're looking at the steps following a cell in suspension interacting with a fibronectin surface. It is too small to see, but I refer you to a guy's review article from 2019. And the time scale on the right shows that this is indeed happening in the second to minute time scale. I want to emphasize here, first of all, the early steps in this process, because it, I think, points out some of the value of the nanotechnologies now in terms of our ability to understand how this system actually works. And it appears that protein clustering is a critical step in forming those nascent adhesions on either a fibronectin or RGD surface. basically we're getting similar activities here. I don't have time to go through all the details here that have been worked out in a number of papers, and the references are below. And we can see that these clusters form, they precipitate actin polymerization, and myosin then pulls on them, and you get a reaction to that, which is further assembly. And this really begs the question of what is this adhesion mutant? And here we benefited from our interactions with Shalom Wind and Mark Schwartzman. And the basic adhesion unit, as coming from his paper in 2010, was roughly 100 nanometers and involved 4 at least RGD units in that 100 nanometers. And this came from fabrication of surfaces with 50 RGDs per micron squared, arranged in either dimers, trimers, tetramers, pentamers. And when he got to tetramers in that roughly 60 nanometer spacing between them, he was able to see cell spreading reliably. We came back to this really in the studies of Rashida Chengidi using super resolution microscopy, and she found that the same size clusters of integrins would form on a RGD lipid bilayer as on an RGD glass surface, indicating that the 100 nanometer diameter here, as seen by the super resolution microscope system, was an inherent feature of that complex. And we wouldn't form larger or smaller reliably. And it seemed even as the cell formed mature adhesions, the integrins were in these 100 nanometer clusters, which indicated that something was unusual about those. And that I'll come back to the possible explanation for that, but wanted to explore this further in terms of what's happening with interactions in 3D, namely with fibers. And we collaborated with Shalom and Yunfei Kai to look at this with 10 nanometer lines and versus arrays of dots. And the 10 nanometer lines, when spaced by 80 nanometers, i.e. they would fit within that 100 nanometer cluster, seams were supporting cell spreading, whereas a single 10, 20, or even 30 nanometer line would not do that. And we see here that the clusters are again the same 100 nanometer in size, and that the constant density of these, no matter whether you have RGD glass or hexagonal dot pattern or a line pair, implies that they also are interacting one with another. And so there's a negative feedback component so that they don't cluster too tightly. But on single lines, they rapidly assemble and then rapidly disassemble in that system. And so we find that the same cluster size is there for the glass continuous, for the hexagons, for the line pairs, but not for single lines and the number of molecules in there. And what we see is that they form on the single lines, but rapidly dissipate because of mechanical forces pulling on them, and whereas the line pairs are able to support those forces, implying that those clusters now show significant interaction between the integrins perhaps facilitated by talin and other integrin binding proteins. The theory that was put forth by Jacques Pro is that it's a question of the curvature, and indeed there's preliminary evidence we have that supports that idea that curving of the membrane by the integrins, and this is something we observed a long time ago when I was back at Duke, that curved surfaces would trap integrins as they diffuse, so they preferred that curved surface. Our further progress in this area really, again, relied upon our engineering colleagues, in this case, Jim Hone and his graduate student, Saba Gassemi. Saba went to the Cornell NanoFab facility and made 9 different arrays of pillars with the geometry down the bottom left, ranging in diameter from 2 microns to a 0.5 micron. And the fundamental difference between the 2 and 0.5 micron pillars was that the larger pillars, the cells pulled to a constant force, but on the small 0.5 micron pillars, they pulled to a constant distance. And from work from Benoit Ledoux's lab, they knew that in the rigidity sensing, it seemed that the constant distance was being used. So you pulled to a constant distance and measured the force as a measure of rigidity. And we went back and now looked at this process of cell spreading, where we saw these periodic contractions that were measuring the rigidity of the surface, and found, lo and behold, that there with the 0.5 micron pillars, we saw pillars at the edge moving inward and pillars inward moving outward. There was a general offset so that the red arrows are larger than the green arrows, which is due to actin flow inward. Since this was pulling to a constant displacement, irrespective of rigidity, and we'll come back to this a little bit later, when we made the surface more rigid, the arrows were essentially the same size because the contribution of the flow was constant and the more rigid pillars were being pulled more strongly by the local contraction apparatus. When we looked at this process in detail over time using nano tracking, something which we actually worked, single particle tracking was something we worked out with Elliot Nelson when I was back at WashU. And we could follow now these pillars as they move toward each other and back. And so very much like the way you and I test rigidity, there was a pull and relax, pull to a constant displacement and measure of force. And this process took less than a minute and then had many downstream consequences. When we looked at where components were there, we saw often a single bipolar myosin bipolar filament between the pillars, and the pillars had alpha-actinin associated with them. Alpha-actinin anchors the actin at the Z lines, and so the idea is you have a mini sarcomere here, and obviously nature knows how to make sarcomeres as the basis for many contractile events and motor processes. So a guy who's on this present and listening went and used a 100 frame per second camera to look at this in detail. And he was able to analyze the position of the pillars to roughly a nanometer and averaging over 15 frames because this was a relatively slow process of contraction. He was able to observe steps. And these steps were roughly a nanometer, and they weren't present in the negative control, i.e. the displacements of pillars outside the cell. And the next question was whether or not when you saw two pillars moving toward each other, whether they were moving simultaneously, i.e. driven by a single contractile unit. Okay. And this is what we observed here. The orange pillar, which was at the edge, was moved inward at the same time as the green pillar, which was 2 microns in, was moved outward. The time separation between the movements is plotted in B here, and we see that that's a Gaussian around 0, and the yellow pillars are not paired and their movements are not coordinated. When we add the total displacement of the pillars together, we see that in the steps, the steps are roughly 2.5 microns, nanometers, which corresponds to the distance that you would move from an actin monomer on one side of the filament, the green, let's say, to the next actin on the other protofilament, the blue, in the background. And the overall distance for displacement for a single actin monomer is roughly 5 nanometers along that given protofilament. So this is a structural aspect of the system. Now, one of the things that became evident when we looked at this and started thinking about this process is that the movements are occurring here with a stiff or a soft pillar. And with the stiff pillars, 2.5 nanometers times the force of 8 piconewtons per nanometer gives you about a significant level of force, which is more than has been generated, at least, in vitro with various myosins. So the question then becomes, how do you get this coordination, and is there some benefit of this coordination? And there we collaborated with Jacques Pro and Francois Ruprecht to try to understand that better. What enabled us to do that was to now UV treat and ozone treat the pillars, and so we can make them more rigid. We increased over by 2 hours of irradiation. We got a 20-fold increase in pillars, and using a EM grid as a mask, we had pillars which were soft, adjacent to pillars that were rigid, and a single cell would span both of them. And so we could look at displacements of pillars at the same time in the same cell on rigid and soft. And the surprising result there was that the velocity of contraction was the same on soft and hard for both contraction and for relaxation of the pillars. And that seemed rather confusing. Okay. And further, when we looked at the peak displacements, we saw that there was a force of about 3,000 or 3 nanonewtons on those rigid pillars, and raised the question of how many myosins were there. So we did fluorophore counting on GFP myosin 2A, which was transfected into a background of myosin null cells that cost 7 cells. and backed out that there was about 50 myosins per contractile unit, meaning that on the rigid pillars, they were generating about 60 piconewtons per myosin head, which is much higher than they had done in vitro. And so Jacques and Francois looked at this process and came up with a model in which they proposed that basically all the heads were bound, and that this was aided by the tropomyosin molecule that was coating these actins, and we'll come back to as necessary for this rigidity sensing. And what happened was that you would have a buildup of the myosins in a high force state, and then a cascade on to the next site on that filament. And this is why you got the 2 nanometer, 2.5 nanometer stepping. It was not because of single myosin was moving by that distance, but at the ensemble average. And this is why you can get such high force per head effectively in contractions. And this is yet to be really followed up on. Getting back to the issue of rigidity sensing, a guy noticed that when you had rigid pillars, they moved roughly 4 steps before they paused, whereas the soft ones moved 10 to 12 steps. And this gave a roughly the same force in those cases when you saw this pause. So that raised the question of, with that constant force, and indeed we saw a similar force for reinforcement with the beads coated with fibronectin, was there a biochemical correlate? And what he found was that when that pause occurred in this right panel, you saw alpha-actinin recruitment to those pillars that were pausing. So there is a biochemical event occurring in that situation, which then leads to further reinforcement. Whereas if that threshold is not passed, you have disassembly and go down potentially a cascade of apoptosis. We can see that in a further study by looking at the DAPK and Paxillin recruitment. Rufang Chin here showed that when you had, again, soft and stiff pillars in the same cell and followed recruitment of DAP kinase 1 on soft and rigid Paxillin, both of them initially started to be recruited to pillars, whether they were soft or rigid, but when you passed a certain threshold, no longer was there recruitment to soft, but there was further recruitment to rigid, and that occurred at the peak of the displacement here when on soft versus stiff. So this raises the question of molecular mechanism. And we and many others have been focused on the process of protein unfolding with force. However, this pause doesn't involve any change in length. And so it seems that something, the force per se, is activating that reinforcement process. One idea that's shown here is that you have the transmembrane proteins connected to an enzyme like PI3 kinase, and that bending of those proteins in the plane of the bilayer takes about 20 piconewtons or so to pull a lipid out of a bilayer. So we're talking similar types of values, would then lead to activation of, let's say, phosphorylation of an inositol lipid, whereas it's not conducive or consistent with a protein unfolding type of model. But this is purely speculation at this point. Looking at the various components and what happens downstream, we know that on a rigid surface, we get talin cleavage and the rod is necessary for growth. On soft surfaces, you also get talin cleavage. The talin head there appears to catalyze or help with the DAP kinase associated death process, which is stimulated by PTPN12. I want to emphasize that this is a general theme. So nature likes to take a theme and modify it and use it for other applications. And we find that there is an analogous cadherin contractions measuring the rigidity of the neighboring cell, which requires that neighboring cell to have myosin 2A in it. So it reacts to that pulling or pinching by its neighbor. And that's involved in epithelial organization. And further studies are being done in a Virgil Vyaznov's lab back at the MBI. The rigidity sensors, as we started looking at the various components involved, appeared to contain many tumor-associated proteins, including the receptor tyrosine kinases, the AXL and ROR2, and the EGFR and HGR2 RTKs are all involved, Src family kinases, DAP kinase and PTPN12 are tumor suppressors, as is tropomyosin. So it appeared that it might have something to do with cancer. So we asked the question of, are they missing in cancer, and what are the consequences of the loss of rigidity sensing? Does this explain the transformed growth, i.e. the growth on soft surfaces? So we looked at 5 different transformed cell lines here from very different tissues and transformed by different means. So +7 is not a tumor cell, but it was SV40 transformed. MDA, MB231 is a breast cancer line, a fibrosarcoma, ovarian, and Lewis carcinoma. And when we looked at the density of the local contractions, i.e. the rigidity sensing events, it was dramatically decreased or essentially missing in all of the transformed lines, whereas human foreskin fibroblasts had it. And when we looked at the various components that we had identified as part of the rigidity sensing machine, we see that in the COS7, myosin 2A is missing. In the MDA, MB231s, tropomyosin 2.1 is missing. The HT1080s, fibrosarcoma, has 2.1, but has tropomyosin 3, which competes with tropomyosin 2.1 and is upregulated in virtually all cancers. The ovarian cancer was missing 2.1, and LLC has a depletion of myosin 2A and a number of the RTKs. So this raised the question of whether or not we can now treat this as a modular system and toggle it back and forth between an active rigidity sensing process and a not inactive machine, which would then stimulate transformed growth. This is an example of doing that with the MDA MB231 cells, where we added back tropomyosin 2.1. The cells in the middle panel here, we see that they pull to the same length, that the rigidity sensors are present, whereas in the control state, they pull much harder on soft than on rigid surfaces, and harder than they do when those sensors are restored, and they don't have any local contractions. but they do grow on soft surfaces, whereas when tropomyosin is added back, they don't grow and they activate caspase here. If we now take a clue from the COS7 cells and deplete myosin 2A, we obviously compromise their ability to contract. They no longer form contractile units, but they now grow on soft surfaces and they don't activate caspase. So it's sufficient now to remove that contractile unit. In other words, not remove a single component, for example, that will block its formation, and the cells will now grow. When we ask the question using RNA-seq, what happens to that cell? Are we just changing one protein, or is it a state change for the cell? The answer is that it's a state change. And we see with the breast cancer cells, adding back tropomyosin changes the level of over 1,000 mRNAs. If we do the converse, if we take the human foreskin fibroblasts and remove tropomyosin, roughly 700 mRNA levels are changing. And so it seems that this is a major state change, and that this transformed and the normal cells are fundamentally different, something which has been evident, I think, in the cancer field for some time. And we can do that with depletion of any one of these 8 different proteins here, will cause the cells to grow on soft surfaces in a transformed fashion. And that adding back those will reverse that process. Many of these are tumor suppressors. So indeed, that state change now is involving the presence or absence of this critical sensor. And if we look at this process of epithelial to mesenchymal transition as a process that is involved in the making more aggressive cancers, we find that, again, rigidity sensing is missing, but various components are missing. About half of them lack tropomyosin 2.1. Of these 20 lines, 1 lacks myosin 2A. A number of them are missing PTPN12 or DAP kinase. And so there are a number of different ways of producing that, but the bottom line is that all of these tumor lines, which were provided by Ruby Wang at Singapore, are missing the rigidity sensing and will grow on soft surface. This raises the question that maybe we can find, as was suggested in the question and answer session, the discussion session before we started this, that there might be drugs or that would selectively target that state. We look for some, and we have some promising candidates, which we may go back to. Or is there some other way of testing that? So we looked at this from the point of view of mechanosensitivity. We had shown before that we could get normal cells to grow on soft surfaces if we simply stretched that surface periodically. And we now tried that with the transformed cells. AJ Taher, a new postdoc in the lab, reported back that when he stretched the breast cancer cells, they were dying, they weren't growing, which was very interesting. We see here the percent of apoptosis, and with the stretch, which is the red line here, we're getting 30 to 40% apoptosis, whereas without stretch on soft pillars, the cells grew fine. However, if we added back tropomyosin 2.1, restored them, now they died on the soft surfaces, and stretch caused them to do better. The mouse fibroblasts started out in that state where stretch encouraged them to grow on the soft surfaces. But now if we subtracted by siRNA, tropomyosin 2.1, made them transformed, they now grew on the soft surfaces just fine, didn't undergo apoptosis, but if we stretched them, they apoptosed. The process of apoptosis here was not the same as the normal cell apoptosis on our anoecus on soft surfaces that's dependent upon death-associated protein kinase, DAP kinase 1, but was dependent upon calpain, and particularly calpain 2 knockdown inhibited it, implying that there was calcium entry. And when we've looked at piezo-1, we find that piezo-1, this mechanosensitive calcium channel, is necessary for the apoptosis process with the stretch. So indeed, it's a calcium-induced calcium response in this pathway. And we see that over roughly a 30 to 90 minute period, a dramatic increase in the calcium levels in the tumor cells with the stretch of this. It's not instantaneous. Stretch is relatively impractical as a cancer treatment. So the issue is whether or not other mechanical perturbations could cause tumor cell apoptosis. One of our colleagues, Felix Margadant at the MBI, suggested that ultrasound would do the same thing. So we tried that out, and sure enough, we found that these are three different cancer lines here, the breast, a melanoma, and the fibrosarcoma. All of them are killed by ultrasound treatment, whereas MCF10As, which is a normal epithelial line, and HFFs are not killed by ultrasound. And in collaboration with John Guinea in Singapore, he made from primary pancreatic tumor isolates tumor spheroids. Those, when treated with ultrasound, died very dramatically. And when we injected the MDA or other cancers into the chick choriontouic membrane and allowed them to grow, these are now a good model for tumor growth. They have all the growth hormones of the embryo. And they typically grow very well, as shown in the control case here. But when we ultrasound treat them, they die in this situation, many of them reverting almost completely. And in more recent studies that are ongoing, we see that in mouse model, we can treat and reverse tumor growth in that mouse model system as well. So this reinforces the idea that indeed mechanical issues are sufficient now to inhibit cancer growth. A good example of this is a paper from Harvard lab where they stretched the mice for 10 minutes a day. The mice had been injected with mouse breast cancer cells in the breast fat pad, and they were able to inhibit growth. If you look at the NCI website, or just Google exercise inhibiting cancer, you find many pieces of evidence of that. The website, NCI, lists 7 cancers where exercise has been proven to inhibit the growth and the severity of the cancer, and 8 more that where it's been implicated. So it seems that this mechanical idea does work. When we look at the details of what happens here, it is this ER mitochondrial interaction, that interface, which leads to calcium-induced calcium release and opening of pores in the mitochondria to release cytochrome C that then activates apoptosis. And how that is coupled now to the ultrasound perturbations, we don't know, or to the mechanical stretching, we don't exactly know. There's a lot of evidence that cancer cells are altered in their cytoplasmic organization. They're softer cells, even though they pull harder on substrates, their cytoplasm is softer and potentially mechanically different in critical ways. So the bottom line here is that blocking transformed cell growth should block cancer growth. And we have indeed found that of over 50 lines, only about 3 are not transformed. These are glioblastoma lines that do have rigidity sensing and don't behave in this classic pattern. But bottom line, over 90% are responding in this way. And we formed a company, Mechanobiologics, to exploit this to develop ultrasound systems that now inhibit cancer aging and injury models. And to give credit here, the initial descriptions of the rigidity sensor from a guy with the help of Giovanni Mecchi. Beverly Lang worked out the RTK involvements. AJ did the ultrasound and the stretch studies. Rashida Changidi on the definition of the organization unit. Rufang on DAP kinase, Yan on the cadherins, and Ming Shi on the piezo-1. We had important engineering colleagues, Shalom Wind and Mark Schwartzman and Hao Gang. All were important in understanding the details of this. Jim Hone and Saba enabled us to get the small pillars that were necessary for this. And we collaborated with Jacques Pro and Jean-Francois and Alexander Bishatsky as well on this. So thank you very much.
Well, bravo, thank you so much, Mike. Oh, there's a an advertisement for a book here. Thank you too soon.
A lot of the basic principles are in this book. So anyway.
Excellent. And who's in the driver's seat of this car before we move on to questions?
The nucleus, obviously.
Mike, can I ask you to stop sharing so we can see the panelists?
Yes.
Okay.
Super. Well, welcome. We've had several panelists join the group, and for those who are new to this format here, one of the really beautiful aspects of this format is something that's made possible almost by COVID-19 is that we're able to actually really have a nice discussion afterwards. And for those who haven't been here before, the conversation always runs pretty long, but it doesn't always get to start with a question from Sam Safran. So it's a special day today. So as you unmute yourself, please introduce yourself, say where you're from, and then looking forward to a discussion. Sam.
Thank you. Sam Safran from the Department of Chemical and Biological Physics at the Weizmann Institute, and I apologize if there will be a siren in the background and I'll have to run downstairs. I have a question, Mike, about the basic science conclusions that one obtains by using these very beautiful pillar experiments. And I, followed that for many years, and Wang, you and others, and Hagai. And that is the following. when you have a cell on a continuum elastic substrate, so let's even think of some artificial substrate type that Dennis uses. The cell stressing one part of the substrate communicates those stresses through the continuous substrate to other parts of the cell. Right. The pillars are decoupled from each other, more or less. And so right. When the cell pulls on a pillar, certainly I agree that you get this very detailed information on the forces exerted locally. But what I am concerned is that you might be losing the more global information on how the stressors are transmitted in the elastic body. And all the engineers here who do mechanics know that these stresses are transmitted in a continuum of elastic body. And you mentioned something at the end, I think that's related to that, which is the rigidity of the cancer cells, or the softness of cancer cells. If you look at pillars, the global rigidity of the cell or of its cortex is not that important because you're just looking what's happening locally. But if you have a continuum substrate, as Dennis, Paul Gianni, and even theory that we did with Dennis many years ago shows, what's important is the ratio of the cell elasticity to the substrate. So my question is there more information that one should get by extending these studies to continuous substrates that might give more global information of how these forces are transmitted?
Well, I mean, the issue is obviously in vivo, it's not a continuous substrate. It's really dependent upon a fibrous organization, which is a modification or a combination of both.
Right, I agree. But those fibers are cross-linked. I agree, but let's just talk about cultured cell experiments.
And I think the other thing that's coming out from some of the viscoelastic types of studies is that when you have a pull and then a creep, you have quite a different behavior because of the temporal components than when you just have an elastic system. Okay.
Yeah, I agree, but that still involves spatial interactions through a continuous medium. I mean, I think the pillar experiments are beautiful because they isolate what's happening in the way that you said, but I think there's more to the story that may be interesting.
There's more. I mean, come on. The point is that we are at the very beginning of understanding the mechanics at the biological level, okay? And that when you think about that bird of paradise and how you form various shapes, you have to have multiple mechanosensitive, mechanoreactive processes involved. And those are going to be, we're saying this rigidity sensor is one. And there are many others, and what you're alluding to is indeed that the process is very complex. Timing, forces, and position, relative position, really do matter a lot. So it's going to take a lot to sort this out. Okay.
Thank you.
Next in queue here is Dennis. Actually, let me see if Dennis is there. Dennis, would you be willing to unmute and ask your question? Super, thanks.
Yeah. So you can hear me okay? Yeah.
Sounds
great. Yeah, thanks, Mike. Thanks a lot for broad view of the molecular field, especially molecular mechanisms. So these two TPMs are especially interesting. I mean, don't have to say that they're highly helical, which reminds me of intermediate filament proteins and collagens and dimers. Stabilizing actin, F-actin is, I guess, at least the simplistic way I look at them. But then there's multiple isoforms and even splice forms. And so there's an interesting paper from Amnon Buxboy and PNAS using single cell RNA-seq, he found TPM1 is particularly mechanosensitive. So it fits with the general idea I think you have. And then I sort of dug into some ENCODE database, and RNA-seq by many other groups sort of fits that. TPM1, maybe 2, but not 3 and 4. Anyways, all of those comments are sort of set up for a question. What do we know about the differential regulation of 1, 2, and 3, and 4, and how should we think about these contributing differently to, I guess, as I say, F-actin stabilization or otherwise? Especially in the tumor context, right. Regulation, dysregulation.
I mean, the important issue here is that there are about 40 different tropomyosin isoforms.
Splice forms. There's 4 genes.
Right.
There's 4 genes and multiple splice forms. Yeah.
They're mostly created by alternative splicing. You're right.
Yeah.
So and the level, according to groups in Australia, is such that all actin in the cell is coated with tropomyosin. That's their implication. And that their idea, in any way, is that the various different actin, let's say, functions within the cell are enabled by different tropomyosins binding them, and that the formins, when they cause actin to polymerize, are co-involved with the tropomyosins, such that the tropomyosins co-assemble on those filaments as the formins are polymerizing. So this view of the world, the cell world, is that the actins are indeed different when they are doing different functions in the cell, and it's driven by the different tropomyosins. So indeed, we don't understand enough about how that actually might occur. And Peter Gunning's lab has led this sort of area of research, and those are basically his ideas, but a lot more needs to be done.
But there is evidence from Amnon, and also, as I say, these databases, that the TPMs are regulated by this SRF pathway, which of course is regulated by G-actin levels. So this at least sort of makes sense to what you're saying, right? That stabilize F-actin in some way, and that regulates G-actin levels, but then the exact calculus by the cell is seemingly unclear at this point. Yeah.
I would say so. I mean, the complexity that I alluded to here with injury repair is that you want the cell to be able to grow, but then to go back to being what it was before. So microRNAs are a good way to do that. You just knock down the level of the growth inhibiting proteins, you enable growth, and then you knock down the level of the microRNA to bring the cell back to normal behavior. And so it's not surprising that there's a very high correlation between the severity of a cancer and the level of microRNA 21. So, it's a good way to do that, okay? And the fact that it targets the tropomyosin 2.1 and P10 and a couple other components indicates that those are important proteins in this growth control pathway.
Okay, thanks.
Ning. Ning.
Hi, Mike. Yeah. Hi, Mike. Beautiful talk. I really like. I mean, of course, this is your last several decades of your work, and especially the recent work on the nanopillars, and then looked at the molecular structures, tropomyosin 2.1 and myosin 2A. and all this is get down to the molecular assembly. I mean, I know you've been proposing these models of modular, units or compartments which can, build together again and again, and then now you put in this cyclical, events. Very, very beautiful. So I would like to ask a general question about this. At the beginning of your talk, you mentioned about this diffusion. Basically, everything's diffusion based mostly, and it's stochastic. And I think Dennis has been using the word called dust to dust, if I quoted him correctly. And I remember I talked with George Whitesides many, many years ago. This is probably more than 20 years ago when I was still working at Harvard. And then he told me the self-assembly is so important. Of course, he's a chemist. And I would like to have your view on this, how we build these units, because I know, and also some of you may have been aware of this so-called liquid phase transition, LLPS. It's been very important in going from liquids to sort of like gels or solid and then back and forth, and controlling assemblies of compartments, especially like nuclear structures, membrane-less structures. What's your view on this? Is LLPS has anything to do or is dominating this process for assembly and disassembly of these microstructures? Well,
certainly the studies of the phase separations more recently from Ron Vale and Jay Groves and others labs indicates that you do get multiple weak interactions, particularly when you activate the RTKs, GRAB2, SOS, etc., form local phases, okay? And then they relax. What is missing here, and what we don't understand, is how we integrate these signals over time to produce the downstream consequences that we want, okay? So in other words, the differentiation process is driven by, let's say, a rigid surface going down a different pathway than a soft, okay? Evidence of that differentiation comes after 4 hours or a day or so. And how many times is the rigidity sensed? How many signals are developed? And what is counting the number of those events? We don't know, okay? But that decision-making process is really critical, okay? And so we know the input, know the output, but what's happening in between is what I'm trying to emphasize here, okay? And the molecular processes are on the sub-second to second time scale. The integrated processes are on the hours, days, even level, okay?
Yeah. If you allow me to follow up on that, a little bit more specific about your nanostructures. You have this 0.5 micron nanopits, and which you said it's your data show basically is displacement dependent. On the other hand, if it's 2 microns or bigger, is force dependent. So why there's such a big difference in this rigidity sensing in the response of the cell at this subcellular nanostructures?
Okay, well, with the 2 micron pillars, they're basically the same size as the contractile units. And what you find is that adhesions tend to form around the edge of the pillars because obviously there are more integrants outside of the pillar top than there are on top of the pillar. And when you look at myosin organization, you find, and we published that in the first paper, essentially a series of sarcomeres around the 2 micron pillars. Okay, so it's forming the same sort of contractile unit, but it's really pinching the top of the pillar, whereas when it can't do that, the pillars are half a micron or less in diameter, then you see this myosin between the pillars and pulling the pillars together.
Thank you.
Thanks. So we actually have a big queue here. It's a great discussion. Thanks to everybody who's contributing to it. So if you wouldn't mind, could you put your virtual hand up? So if you're retired, you don't have to find that. But if you're not retired and you have a question, please go to the raise hand function, which is just to the right of two slots to the right of your share screen option. So the next person in queue is Shreyansh. And Shreyansh, be sure you unmute and introduce yourself.
Yeah. Can you hear me?
Yes.
Yeah, very interesting talk, Mike. I would rather put a very simple question, basically very fundamental indeed. So basically what you mentioned is all the mechanotransduction, which happens through the focal adhesions and which helps eventually in the rigidity sensing based on how stiff or soft the substrate is. But what parallel I see in terms of when you apply a cyclic stretching, realized either by ultrasound or by stretching PDMS membrane, elastic membranes. These two are a bit different in terms of mechanotransduction, which happens eventually in the system. So what exactly bridges, in fact, a magical bridge, which kind of binds these two and realize the same effect, but with completely different organization of stimulus?
The short answer is I don't know. We're trying to find out what might be the connections there. As I alluded, the mitochondria ER junction appears to have a major role in that. And what has emerged really in only the last 5 years in detail is that many of the internal membranes in the cell are mechanically coupled. That's critical for transfer of lipids between the site of lipid synthesis in the ER to mitochondria, to plasma membrane. In fact, one of the ways of targeting many of the parasites that we get, like Leishmania and so forth, is to target that lipid transfer process, because it's absolutely essential for cell growth. And obviously it's mechanical in nature, and how it might interact with the actin cytoskeleton and various actin subpopulations is perhaps a major component in that, but we don't know in detail.
So like maybe we can can we expect even going upstream a more unified signaling, which is completely dependent on some X protein membrane bound, which gets affected by these two different pathways?
Well, I hate to say universal, but indeed it's very robust from various different systems. Okay, and it's a highly robust process, so consequently, you might call it universal. Yeah.
Thanks.
Thanks. Excellent question. Next in queue, we have, sitting in front of a bright and beautiful day in Philadelphia, Pennsylvania, Paul Janby.
Hey, Paul.
Oh, hey, Mike. Hey, so what sets the time constant for the rigidity sensor, and could that be altered?
I mean, the study by Beverly Yang in collaboration with Sasha on the AXL and ROR2 RTKs indicated that ROR2 would knockdown of ROR2 would increase the time of the contraction by a factor of 2. So in its ROR2 interacts with filament A, And filament A knockdowns, when we look at them in terms of the pillar contractions, causes abnormally fast relaxation of those pillars. So it's tied in with the process of pillar anchoring to the actin cytoskeleton, and in some way is controlling those mechanical properties. Yeah. So ROR2 interacts with filament A and binds to it and recruits PTPN12, which PTPN12, when we increase its concentration or activity, causes more rapid relaxation of the system.
So you might expect those perturbations to change the way that the cells respond to these viscoelastic, these dissipative things. Has that been looked at yet?
Oh, yeah. Not to my knowledge. Okay. I mean, the kinase phosphatase pairing is clearly a critical issue in terms of adhesion protein lifetimes.
Great question, Paul. But before we move on to the next question, I just want to highlight a note from Genin, which is that number one, I miss all of you guys, and number two, we've paid a lot of money to Elsevier paid a lot of money to Zoom for really great bandwidth. Please feel free to turn on your video. I'd love to see all of you if you're not running to an air raid shelter or in a part of the world where you're in your pajamas. Anyway, the next in queue here is Yingxiao. So Wang Laoshe, please introduce yourself, let everyone know where you are, and looking forward to your question.
Thank you, Guy. Great seeing you, Mike. It's I'm a professor, actually, for those who don't know me, at UC San Diego in bioengineering. So I'm really, thrilled to hear Mike's beautiful talk and work as well. So it's inspiring to know this cancer cell has a switching kind of mechanism for the rigidity sensing. I'm wondering whether, as you also point out, nuclear perhaps is a driver for this whole system. Have we looked into, this genome architecture, how this cancer cell and the normal cell, comparing whether there's a rewiring, mechanism, particularly, let's say, whether there is, physical coupling between the chromatin structure, subtle structure, and the genome loci, specifically for cell growth and apoptosis. I don't know whether you have looked into them, whether there is a rewiring mechanism or effect occurring between these two types of cells.
Well, in fact, we are in the process of doing that. My next door neighbor through that wall is looking at genome organization, groupings within the nucleus using high-C and other approaches at the nanometer level to look at gene apposition and how that might change when we transform cells is the question that we're asking. The samples should be going for the high-C analysis within the week. So,
how about the physical structure and the genome interaction, for example?
Yeah, that's basically what they're looking at in that. So the high-C is obviously involved in the cross-linking of various DNA components, and from the organization that they can back out, they're defining regions which are condensed in one nucleus versus another under different conditions. So indeed, that's what we hope to obtain from those type of data.
Thank you.
Thanks so much. And Amber, before you go, I'm actually going to insert Sulin. Sulin's raise hand function isn't working, so we're going to go from Wang Laoshe to Zhang Laoshe. So Sulin, please go ahead and jump on in.
Hi, this is Sulin from Penn State again. Mike, very convincing talk on mechanical process. So that means that mechanical stretch can suppress tumor growth. But a couple of years ago, I have spotted a paper published in Nature that saying that mechanical stretch can increase the metastatic potential of even genetically primed cells. Do you see any potential conflicts of these two studies driving the different ways? Do you have any comments on that? And by the way, I think that Taher Saif, a couple years ago, maybe 5-7 years ago, he cultured cells on the substrate, stiff and soft substrate, and he found that the same thing happened. That means that if you have a stiff substrate, the cells metastatic potential increase. So I'm wondering if I have any comments on these studies, the increased metastatic potential and your studies.
So I think that one of the issues here is the magnitude of stretch and frequency. Both of those matter critically. If we take the normal cells and we stretch them 15%, it's causing damage, and there are definite reactions from that. In the case of the cancer cells, we're typically stretching them in a physiological range of 3 to 5% strain is sufficient to fully activate their apoptosis. Okay, and so it is indeed in a low level that we're seeing this, and we're doing it at frequencies of basically 0.5 hertz. So stretch for 1 second, relax for 1 second. And once you get out of the various time ranges, you have different effects. Okay. So the details there do matter. Okay. And as I say, we don't fully understand what that necessarily means. And going back to the issue that we feel this is driven by ER mitochondrial stress pathways, and we don't know how the mechanics of those are governed, we have very little that we can actually say about what's going on.
Thank you. It seems that the direction, magnitude, and duration of the mechanical stretch, or mechanical forces, is important here.
Yeah, it is indeed.
Thanks.
Excellent. Amrinder, could you unmute yourself and introduce yourself?
Hi,
Just introduce yourself to the whole gang here. 100 people online still.
Absolutely. This is Amrinder from Mechanical Engineering at Virginia Tech. I work with cells and understand how cells interact with fibers. Mike, a really nice talk. So I have a question for you regarding your first part of the talk, where you're talking about the integrin units being about 100 nanometers in size. And this follows up with what Sam was saying, that when we look at the cartoons, these are all uniform substrates, 2D substrates or pillars. But when you look at the cartoons, the lipid bilayers are all continuous, flat, right? What happens if you were to have high curvature, because you alluded to curvature effects?
Sure.
If these cells were cultured on very small diameter fibers, because when we culture them on 100 nanometer diameter fibers, for example, we don't see any cells spreading. The cells remain rounded, and they're actively protruding because they don't have enough surface area, or the curvature is too high and the membrane is too stiff, or maybe it does not allow these. Do you have any thoughts on how this fundamental biology would extend to a 3D curvature-based environment?
I mean, the difficulty that we had in terms of the initial studies on this was that it was fine to fabricate 100 nanometer, or 10 nanometer or 20 or 30 nanometer lines on a flat surface, ask questions about that and their organization and so forth. But it was much more difficult to move this into true 3D, although we tried some initial studies with Sam Stoop, in his fibers to look at that. The point you raise, I think, is important. We see when there are 10 nanometer fibers crossing like so, that adhesions form on those sites. However, if we have a straight fiber, we have to go to about 40 nanometers in fiber diameter, 40 nanometer width of the RGD before we can get adhesion to it. And with your 100 nanometer diameter fibers, I'm not sure that there is a flat region large enough to enable that adhesion formation. And the direction of curvature that we expect for the integrins, they, in initial studies with Rick Horowitz and his graduate student, I'm having a senior moment here. Anyway, she's now in Texas, a faculty there. She showed that the integrins were diffusing along the edge of the cell so that they liked that outward curvature. And so when you have binding to a fiber that has this curvature and the integrin clusters are going to form an outward curvature, you're counterbalancing that, and it would be expected that there would be that type of curvature would be detrimental to forming a proper adhesive contact.
So I would, if I have a follow-on, just a quick. So I would always imagine of it, because I don't have an experimental proof to show, but I always imagine it to be the membrane of the cell applying a force, because as it's trying to bend, and then you have also integrins that are trying to latch onto the fibers. I think there is a lot of competing balance going on between the forces. So probably integrins are being latched on, but then being pulled off. It's just a hypothesis, but then I also think that they might be also aligning along the fiber axis. There must be some sweet spot at which contractile machinery is enabled from the initial adhesions that form, which is quite dramatically different, I envision, from what's happening on flat 2D or even on the pillar systems.
Well, that's what it, I mean, our interpretation with the 10 nanometer and 20 nanometer fibers is that the integrins initially accumulate there, and when they activate actin polymerization from that cluster, myosin pulls on that, or the general flow of actin interacts with that, and you get forces on those integrins that they can no longer sustain by that simple clustering. Okay. The other thing that I would hasten to point out, this is going back to my days as a postdoc, the bilayer couple is critical. And we understand relatively little about the relative pressure in the, or tension in the outer half versus the inner half of the bilayer.
Right.
And there's many reasons to believe that piezo 1, for example, is responding more to that than to the overall tension in the bilayer, but that differential inner surface versus outer surface tensions may have critical roles in controlling those channels.
Thank you.
Thanks. So actually, what happened? We lost Tang Tian. Tang Tian was actually in. She's gone.
Yeah, well, I lowered my hand. Sorry, because I think my question is last asked by Suling and Yash. But maybe I'll just repeat my question. This is Tian Tan from University of Alberta in Canada. So it's really intriguing talk, and I'm happy to see this interesting connections between mechanics and biology. So I was going to ask whether you think it matters how the force is applied on the cells to cause apoptosis. And I think Sulin's question was related to that, so that's why I lowered my hand. But my question is essentially in the control experiments, you are stretching the substrate, and in the ultrasound, I think perhaps the loading is more complicated, and the substrate is likely very anisotropic, and your cell is very anisotropic. So how it matters, you know, whether you apply a force in stretching mode or in shearing mode or combination. So that was my question. Thank you.
Sure. Well, I mean, there's a lot more to say about that issue. The implication from our studies with the ultrasound-induced killing is that it doesn't matter where the cell is. It doesn't matter whether it's in a tumor, in a tissue, as in those spheroids, or in on a surface, or there's some implications that even circulating tumor cells are mechanosensitive to increased fluid shear and so forth. A number of studies have been published on that. So we think that it's the internal membrane systems that are being affected. And the analysis of how is ultrasound interacting with cells imply that the membranes are indeed an important aspect, and that they're getting sheared, and that there may be changes in just what I'm talking about, namely the membrane tension that are driving these changes, thereby opening channels or causing alterations there.
Thank you.
So thanks. Amir, you're next in line. And could you introduce yourself to the gang first? Great to see you here.
Thanks, Guy. So my name is Amir Zatpour. I'm professor of biomaterials at Delft University of Technology in the Netherlands. And so given my background in biomaterials, one of the things we have been trying to do is to use pattern surfaces, nanopattern surfaces, to kill bacteria. That's what we try to achieve. And so there is a body of literature on that. And then there is this other body of literature which looks at curvatures of you know, a few hundred microns and how that affects the tension in cells. And both of these two seem to work through tensile stresses in the cells. So you basically stretch them, and you might be able to kill them, or maybe you can cause them to organize differently and produce the extracellular matrix differently. And so what is often mixed in these pictures is the effect of geometry with the effect of rigidity.
Sure.
And so, you know, there was recently, we have seen it, others have seen it, that, you know, how much water you put in your test, you know, how much medium you put in your test dish makes a lot of difference in how much cell viability you get, because you regulate the amount of cell stretching by imposing certain gravitational forces. And so, do you think this does it make any sense? I was, you know, I don't really know the exact setup of the experiments, but it seemed to me like some of these experiments might be having both of these two effects of mechanics and geometry, or am I mistaken?
Well, I mean, I think that the obvious implication from biology, going back to biology and the biodiversity that's out there, you know, nature must have ways of understanding and developing the proper curvature on a nanometer level. And, you know, how these systems develop within the organism are critical for organism survival. And so they've obviously come about through an evolutionary process and are refined by that process. So what I think we have somewhat naively assumed is that essentially one size fits all, and that this is going to be, you know, when we look at one cell over here, we'll understand what's going to happen with that other cell over there, and we'll understand all these different processes. And I think one has to really get down to the nuts and bolts and say, if you've got a surface like this and the cell is pulling across that, obviously the separation of the actin filaments from the membrane is going to be greater across that distance than if you have a completely flat surface. That will have implications for the dynamics of those adhesions, et cetera, et cetera. And once you get into the details of it, and dynamics are a critical element, I think one can start to understand these things.
Yeah, exactly. So I think at least we did some studies on that, and we could really never separate geometry from mechanics. So stretching, because you apply certain stretches, and you know, even in the nanometer range, there is a big difference mechanically between 10 nanometer and 50 nanometer and 150 nanometer. It's the cells start to stretch very differently by themselves, and also the anchorage imposes certain and also the material, so how much deformation you get from your material. So I think, and there are certain second order mechanical effects in severe deformations that make it really complex for us to separate the geometry from mechanics. I think that might be worth looking a bit more into. What part of that is pure geometry and part of that is mechanics?
I mean, I admit that we've taken a very simplified view initially, looking at repeated patterns at the nanometer level as a means of avoiding, or trying to decrease the number of variables. Okay. And also repeated patterns enable you to see and to average over a number of different circumstances and see if the same behavior in this portion of the cell occurs in that portion of the cell. So we have the capabilities now with the nanofabrication methods that are out there to start to do this better. Yeah.
Thanks a lot. Appreciate it.
It's the question here. So next in line is somebody who's been waiting almost literally all day for this question. I think it's 11:45 in Singapore right now, 11:45 p.m. Is that right, Andrew?
Yeah, that's right. That's right.
Could you reintroduce yourself to the gang?
Sure. So for people who weren't here earlier, my name is Andrew Hawley. I'm a PI at the Mechanobiology Institute and in the NUS Biomedical Engineering departments. So thanks, Guy. And hi, Mike. Thanks for your talk. Hello from Kent Vale. So I was kind of curious, based on what you've shown, I would kind of assume that tropomyosin deficient cells, which can't really sense stiffness so well, would be susceptible to this mechanoptosis in a stiffness independent fashion. But I'm wondering if you've tested this by plating 231s or transform cells or other cancer cells on soft versus stiff sort of substrates, or even in confinement, to see if there's any difference in the ultrasound killing.
The simple answer is that, yes, there does appear to be greater killing on soft surfaces than there is on rigid.
But as far as like a mechanism for that in cells that maybe can't sense that stiffness?
Well, I mean, it's clear from looking at the soft versus rigid pillars with transformed cells that they pull to greater forces on the greater displacements, I should say, on the soft than they do on the rigid, although they probably pull greater forces on the rigid. So it's in that realm that I would say it's worth looking to see what might the difference be.
Okay, cool. And one minor follow-up, are there any classes of cells in vivo that are just naturally deficient in tropomyosin that might also be sensitized or at risk to an ultrasound-based therapy?
Well, to my knowledge, no. I mean, I think that the rigidity sensing process appears to be a critical one from all that we're doing in regulating growth. So in the adult organism, if a cell falls out of a tissue and is on a soft surface, you want it to die. And so consequently, you want to have that means of activation of apoptosis.
Sure.
So the other thing that I forgot to mention, which I think is fundamental here, is that for ultrasound-induced killing, myosin 2 needs to be active.
Mm-hmm.
And greater myosin 2 activity, as was produced by, there's a paper we have now in press, soon to be in press, on inhibitors of the cell cycle and seeing if there's cell cycle dependence to the ultrasound-induced killing. And there really wasn't much of an effect, but there was a dramatic increase if you added microtubule polymerization inhibitors, which would increase myosin contractility and consequently increase the killing caused by ultrasound.
Awesome. Thanks, Mike, and thanks for the bird of paradise picture too.
I think we worked hard for that one. Farhan was next in line, but where did Farhan go? Farhan.
Yes. Hi, Guy. Yeah. So part of my question was answered. It's a great talk, Mike. So I'm Farhan Chaudhry. I'm from SIU Carbondale. So my question is more about in vivo tumors. If you think about stretching those tumors, there are presence of, I would say, cancer stem cells or cancer stem cell-like cells. So we have published in the past that those cancer stem cells lack adhesion, they don't spread well. So if you are stretching those tumors, in vivo tumors, then there's always a possibility that they may not be effectively stretched. And then there's also a possibility that the primary confinement may be broken, right? So you are unleashing kind of like a more metastatic possibility. So I'm not sure how that's going to work out, but any comments on that?
Okay, well, Peter Friedl has the idea that you don't want to deform tumors, that will cause metastases, etc. And certainly one of the early, shock therapy for tumors and focused ultrasound, all of which lead to heating and localized damage. Those methods tend to activate metastases. Okay. So the issue of deforming tumors in vivo, we've sort of moved away from. There is a intense massage therapy in southern India that's supposed to have beneficial effects for cancers. I haven't followed up to understand the details of that. But anyway, there's lots of jokes we can, make about this sort of thing. Anyway, the importance of the ultrasound, however, is that we're using frequencies in the low range that penetrate the body, are absorbed very minimally, no heating occurs, and there's no damage to the normal cells. And it seems to be relatively independent of adhesion, etc., organization of those cells.
Well, that's a great question. Next in queue is Chen Laoshe from Zhejiang, staying up to the middle of the night. Thanks so much. It's great to see you, Jack.
Hey, hi, thank you. Hi, Mike. Hi, Mike. Good to see you again. Yeah. So very nice talk, and so many friends here. I just have two questions. First of all, I just very got intrigued by the idea use ultrasound to kill tumor cells. So my first question is, how would the ultrasound achieve high selectivity to kill only tumor cell instead of normal cell or other, like immune cells in the tumor microenvironment?
As near as we can tell, the calcium homeostasis pathways in normal cells are not altered by the ultrasound. Whereas the different organization of this ER mitochondrial stress pathway in tumor cells causes it to be particularly sensitive. Now, you can look in the literature and find that a lot of natural products actually will cause tumor cell death by activating a calpain-dependent calcium release in those, and that the cells are sensitive to that pathway. Okay.
I see. I see.
There is generally overexpression of calpain, high levels of calpain in tumor cells, and the calcium pathway is important for tumor growth, which may make it then sensitive to, let's say, overstimulation under those circumstances.
Right. But I'm just curious. Ultrasound has been widely used in a clinical practice. So why this way to kill tumor cell is not that well, like?
Typically, they're using much higher wavelengths because they're doing imaging.
Okay.
We're not doing imaging.
I see.
We're using much lower wavelengths.
I see. A different.
Yeah, and those are interacting with the cells in a different way.
Okay, interesting. Okay. Right. Another question is regarding the liquid phase separation that Professor Ning Wang asked you. So I just want, because liquid phase separation has been kind of very hard recently in a lot of fields, both biophysics and cell biology. So what do you think about whether the LLPs, PS, could regulate mechanical transduction in cells from mechanical sensing on the cell membrane to nuclear, to the gene transcription in nucleus?
Right.
Because it's a kind of build up the kind of membrane-less compartment. Okay, it's very different from this traditional recognized like type signal transduction using like soluble form of molecules.
Right. I think it's important to understand that formation of a community, so to speak, of molecules is what we're talking about here. The consequences of that, in other words, how do you get back to this integration issue, I think are not obvious. And whether that localized aggregate and obviously the stimulus that comes from that will have long stream consequences for the cell really means that something either in the nucleus or in the other components which are decision making has to occur. Okay. But I think it's a critical element. Yeah.
I see. Okay, thank you very much, Mike. Great to see you again.
Great to see you.
Next in queue, we have Li Cheng. Do I remember correctly that you're in Europe right now, or are you back in Singapore?
Me?
Yes. Where are you right now?
I'm in Paris.
Yeah. Très bien. Magnifique.
Thank you.
Please introduce yourself to the gang.
Thank you. Yeah, as you know, I graduated from Singapore. I graduated from NTU Singapore in 2016. So now I'm a contractual researcher in Institute of Global Physics in Paris, France. Thank you. Thank you very much, Mike, for giving this very nice talk. Actually, I'm not in the biology field. I'm in the crystal deformation, the physics field. So I'm just curious about the stretching function working on the cell structure. So from my experience, so if we, for the normal material, so if we stretch the material, then it could cause the structure change. But if we applied some other functions together with this stretching, then the structure change could be decreased or increased. So I'm just wondering if we applied other function factors as well together as with this stretching, it could also change the cell more or less, especially for this kind of biological material, you know. Thank you.
Sure. I mean, I would go to what my initial reaction to that is that indeed the result with the myosin inhibition is relevant. So if we inhibit myosin contractility, we know we change the organization of the cytoskeleton, and that is sufficient to block the ultrasound-induced killing of the cancer cells. So it requires the organization of the cytoplasm driven by myosin and downstream actin polymerization. The two are coupled in most cells for this to actually happen.
Thanks. And then,
thank you.
Thank you, Ching. Thanks for being here. And then
Thank you, guys. Yeah.
Trop bien. Merci, I mean. So we now have, we're coming close to our time here, but very pleased to have another question coming all the way from Singapore. So Sunu Zhe, could you unmute yourself and introduce yourself to our, oops, there you are.
Oh, hello. My name is Sun Yuzhe, and I'm from National University of Singapore Mechanobiology Institute. Thank you, Prof. Sheetz, for your excellent talk. My question is about the tropomalsin's function in cell rigidity sensing. Because the tropomalsin 2 and tropomalsin 3 actually have a very similar sequence, although we didn't know the exact structure of these two proteins. I wonder, what is the mechanism that causes their totally opposite function in terms of the cell rigidity sensing? Did you explore this detailed molecular mechanism, or do you have any thought of how can we tackle on this question? Thank you so much.
Sure. I mean, there's evidence from Peter Gunning's lab, which we indirectly confirm that tropomyosin 3 will activate myosin movement on actin. So actin coated with tropomyosin 3 will catalyze a higher myosin ATPase activity than that coated with tropomyosin 2.1. Okay. So, and that presumably has to do with the interaction site, secondary interaction site between myosin and tropomyosin. And we know that tropomyosin 2, for example, depletion will increase the velocity of pillar contraction. So indeed, it has consequences in that activity realm. And the issue of the competition between the two, we don't fully understand, which would get back to the logical hypothesis, let's say, from Gunning's lab would be that the formins will choose both of those for addition to actin filaments. And so therefore, an overexpression of 3 will lead to 3 coating on the filaments and those don't participate in the organization of the contractile units for rigidity sensing, it seems. A lot of this is hand waving at this point. Okay.
Thank you so much.
Great question. We're coming up to the end of our 2.5 hours here, but there is one more question that I want to make sure gets answered. It's a really nice question that was asked in the chat in the Q&A, and unfortunately the person who asked it left before I discovered that the chat function existed. But it's a really great question, and just in case he comes back to see the YouTube video, it's kind of a big picture question. So when you're, I'll paraphrase this question saying, when there are passive and active components that contribute to mechanosensing and to force sensation and to structure of the cell. When looking for targets for mechanoapoptosis and for ways to really get external energy into a cell and make it do something, is it really more the passive or the active elements that are your target?
Well, obviously, from the dependence on myosin contractility, active components are necessary. The issue that you raise, I mean, gets back to the whole matter of exercise and its benefits. And clearly, you are deforming the system. The system is getting signals from the outside which activate it, okay? And it's that stimulus of activation, presumably, that's a critical element in the downstream processes. In other words, inactivity begets further inactivity. You lose it, you use it or lose it, okay? And I think that as an overriding principle in biological systems is important from, I mean, it's logical from an evolutionary viewpoint as well. So when the organism reaches a stage where it's no longer actively engaged, why keep it around? So you activate the apoptosis process and activate death as well, senescence and death.
That is an absolutely fantastic thought to reach the points in our conversation where we all get up and get a little bit of exercise and walk around. But I'd like to thank you, Mike, for bringing this entire group together. Thanks for a great talk. It's wonderful to see all these faces from around the world, people I haven't had a chance to interact with since before the pandemic, a time I hardly remember. It's been a wonderful discussion, and thank you for leading us through it, Mike.
Well, thank you.
Can I, before you leave, can I just show maybe a couple of slides? I have actually quite a few questions, but I didn't have a chance to answer, to ask. What I want to show is the next week's EML webinar. Let me see if I can find that slide. Where is it?
Jimmy, it should be the last slide of the.
I know, but let me see if I. Here. Okay. By the way, let me repeat what or just mention what Tong mentioned. This GoFundMe. We just got some feedback that maybe some people in different countries would not be able to reach that site. So take that, we'll keep that in mind and maybe change that. And the announcement is, first of all, thank you, Mike. It's a wonderful, wonderful talk, as usual. And I also like to thank Guy and thank Taher for leading the discussion. Beautiful discussion. And thank all the participants. And next week, next Wednesday at the same time, we'll have Edward Arst giving an EML webinar. I'm looking forward to seeing all of you. Thank you very much.
Thanks, Jimmy. And Mike, thanks again. You continue to make Washington University proud.
Okay.
Thank you.
Thank you.
Yep.
Thanks, everyone.
Thank you. Bye.
Bye-bye.
Bye. Thanks, Mike.
Jimmy, thank you.
Thank you. Have a great day.
You too.